3.5 billion years ago, organisms lived which left fossils. And so we know that as of that point, planet Earth had life. But this life was prokaryotic cells. Even though today Earth's life is full of eukaryotic cells, big complex cells, which include animals, plants, and fungi, the first life did not include any of these complex eukaryotic cells. Rather, all living things were simply prokaryotic cells, which in general tend to be small and simpler, lacking a nucleus and membrane-bound organelles. About 2 billion years ago, a different type of cell appeared on planet Earth, which were larger and had nuclei and other membrane-bound structures. These are human cells of the intestines, here's a white blood cell, the bone marrow, another bone marrow cell, and here's a nerve cell. Notice that these are larger and have a nucleus. And so these are different from those prokaryotic cells. And so if you were to look at the history of life on Earth, for a while, there is no trace of anything alive on our planet, starting with its origin 4.6 billion years ago to around, once again, 3.5 billion years ago, maybe 3.8 billion years ago. And then there was life on Earth, but the only life was of the prokaryotic cells. So bacteria had planet Earth to themselves for about a third of the history of life. Now, eventually in the Phanerozoic eon, there would be vertebrates and there would be plants. Prior to this, there would be invertebrates. Fungi are a little harder to identify simply because some fungi are unicellular and some fungi, and then fungi typically then have these microscopic strands known as hyphae. And so while plants and animals tend to be larger and multicellular, fungi are found in the fossil record, but a little harder to trace their origins. But prior to any animals or plants, there were eukaryotic cells, which we call protists. And protists, as I'll say, is not a good biological term, but it's the term which can be used to describe eukaryotes, which are not plants, fungi, or animals. And they existed for about 1 billion years or more before the first animals, originating, say, 2 billion years ago, and animals not appearing until, maybe 700 million years ago, give or take. So eukaryotic protists, they're exciting. Not only are they exciting because they're important in natural environments, but also because we are eukaryotes. Animals are composed of eukaryotic cells. So the first eukaryotic cells were significant. Now, we call ourselves eukaryotes because there are human cells, sperm, which have flagella, just like these swimming protists have flagella. Now, it is true that bacteria and archaea have flagella, but they have two different kinds of flagella. So the archaea of the archaea and the bacterial flagella of bacteria are completely separate structures from those of eukaryotes. But there are some fungi which have flagella. Plants can have flagellated sperm, as can animals, and as you can see, these protists. And so eukaryotes share this feature. Eukaryotes share the feature of having a nucleus of membrane-bound organelles like Golgi or endoplasmic reticulum, of having mitochondria. If you look at how this amoeba is moving as it dissociates its cytoskeleton and then reassembles it, bacteria can't do that. They don't have a cytoskeleton composed of actin in this way. Bacteria don't have cilia, which are composed of the same microtubules as flagella. Only eukaryotes have microtubules and cilia. And so whether it be cells of the human throat or cells of this one-celled protist paramecium living in a pond, these cilia are similar structures. Bacteria divide through fission, whereas eukaryotic cells have a more complex cell cycle involving mitosis with prophase, metaphase, anaphase, telophase. The point is that from the generalized living things came a subset known as eukaryotes, and they share features because these features evolved in the earliest eukaryotes. And then the groups which evolved from the eukaryotes, the fungi, the plants, and the animals, then retain ancestral features. So finding eukaryotes is in the fossil record. It's significant, not just because eukaryotes and protists are important, but because here we're seeing the origin of the foundations for human structure. So for example, the actin, which composes the cytoskeleton, and the myosin, which can pull on it. Humans and other animals will reorganize to make muscle with actin filaments and myosin pulling on that. And so the origin of the first eukaryotes is significant. Now, one could ask the question, where did eukaryotes come from? So if the world was full of these small, simple bacteria, and now 2 billion years ago, there's big, complex eukaryotes with nuclei and mitochondria, where did they arise? Well, here's where we just kind of sort this out, where it is easy when, especially, working with, students who want, in a prehistoric life course who want to talk about dinosaurs and not so much, fossil cells, or when dealing with, people who are beginning their study in biology, to generalize. So bacteria, let's, simplify them. They're small, they're simple. That is generally true. However, bacteria are amazingly diverse. They compose 2 kingdoms of life. And so while many of them are about a micron in size, that is 1,000th of a millimeter, and many of them are smaller still, maybe half that size or even smaller. There are some which are much, much larger. In fact, there are some which are so large that they can be seen with the naked eye without a microscope. So in this animation, the smallest bacteria are these small ones, right? And you'll continue to see those. Those are the dots here. This larger square-shaped bacterium, this is its actual size. This rod-shaped bacterium, that's its actual size. So see how much larger they are than the small dots. And then there are some which are are larger still, which one can observe without a microscope. So the point I'm trying to make is, while it's appropriate, to just generalize and say, on average, bacteria are small and simple, then that raises the dilemma: how could a small, simple cell become this big, complex eukaryote? When you realize that bacteria actually can be large. There are bacteria like these which are larger than many. In fact, this one, most eukaryotes. So some eukaryotes are rather small. Some bacteria are rather big. So there's an overlap. So it's not as if we're dealing with, things which are completely different. And then this also applies to other aspects. We do not have to, get into, all of, the weeds here. But while bacteria in general, we said they have a circular chromosome, eukaryotes have a rod-shaped chromosome which can undergo mitosis. There are bacteria which, instead of having circular chromosomes, have linear chromosomes. So some bacteria have, quite large, genomes, have rod-shaped chromosomes. Some bacteria actually wrap their DNA in a membrane. So once again, something I just said was bacteria don't have a nucleus. The eukaryotes do. However, like I said, there are some bacteria where there is a membrane around the chromosome. We don't call it a nuclear membrane, but nevertheless, it's an internal membrane. There are some bacteria which highly fold the cell membrane, such as the cyanobacteria and this Epulopsychium. The cell membrane is highly folded, so it's not an internal membrane-bound organelle, but it amounts to about the same thing. And so, for simplicity's sake, it's easy to say bacteria, small, simple, eukaryotes, big, complex, but that then raises the question, well, how could eukaryotes arise from such small, simple cells? And the reality is that not all bacteria are, so small and so simple. Now, last I checked, we're not quite sure where the nucleus came from. All right, and so here you see the DNA is wrapped in a membrane. Here in this ovum, the DNA is wrapped in a membrane. And so somewhere in fossil eukaryotic organisms, that was invented, a nucleus where all of the chromosomes are wrapped in a membrane. We're not sure where that came from. It may not be that complex. Some bacteria already wrap their DNA in a membrane, so maybe it was just an elaboration of something the bacteria already had. Another possibility is that cells began to live with inside other cells. And so the idea that now we have a membrane-bound nucleus inside a larger cell, that might be the result of cells actually fusing. Now, well, there's definite evidence of this coming up soon. But one of the reasons to suggest this is that there were two different kinds of bacteria. There were the archaea and the eubacteria. And if there are two bacteria, you could ask, where did the eukaryotes come from? Did they originate from this one or this one? And the fact is, certain genes suggest that they came from the archaea. They're far more similar, eukaryotes are far more similar to the archaeal genes than the eubacterial genes. But then other genes, it's the similarities with the eubacteria. That makes no sense. How could it be both? And the idea that now two cells have fused would then explain how you could have eukaryotes with similarities to these two different organisms. So how the nucleus arose, there are different models. Some even suggest that a large virus might have been involved. And last, I had read, it's difficult to test. However, the second largest structure in the eukaryotic cell is the mitochondria, or in some chloroplasts, but I'll get to those as well. And so we could then ask the same question, and here there is much better evidence. So eukaryotes, in addition to having a nucleus, which is their largest organelle, have then mitochondria and chloroplasts. Where might these have come from? Well, eukaryotic cells today commonly eat bacteria. All right, so amoebas and paramecia, they can ingest these bacteria like blue-green algae or other bacteria, and this is then their food. They secrete enzymes, they digest this, they absorb the good stuff, etc. So a cell can be taken in from the outside, be incorporated into a membrane bag where it is digested or food. There's a process here known as phagocytosis, or cell eating. Okay, but there are variations on this. So first off, sometimes the smaller cell wants to get inside the big cell because it's a parasite and it takes over. So in addition to the relationship where one eats the other one, there could be a relationship where the little one benefits because it's parasitizing the big one. But there's also a third possibility where one now lives inside the other and they both benefit. A relationship where one cell lives inside the other and they both benefit is called endosymbiosis. Symbiosis because they both benefit as opposed to, say, parasitism. But then endosymbiosis because one is actually inside the other. And so if this became a permanent condition where this small cell is now living inside the big cell, it is, and they are both benefiting, it's what's called a symbiotic relationship and an endosymbiotic relationship because now one is inside the other. Okay. Now, when this was first proposed, it wasn't taken all that seriously because it seems a bit absurd until it was realized that this happens quite frequently. There are many examples of endosymbiosis which are known. So I just wanted to kind of mention um, as some of the ones in um, which are easy to identify. So for example, living things can associate with other living things in a relationship where they both benefit. So if you were to say, look at the egg masses of wood frogs, very often they are green. And if you were to ask, why are they green? It's because the animal embryos are developing with green algae and they both benefit. So the embryo is producing carbon dioxide and nitrogenous waste that the algae likes. The algae is producing oxygen and perhaps even sugar, which benefits the embryo. So that is a symbiotic relationship. If you were to consider spotted salamanders, it goes even further because the green algae actually live inside the cells of the salamander. So once again, the embryos and the egg masses are green. There are actually algae living inside the cells of the animals. And so this is a symbiosis, a relationship where both benefit. And it is an endosymbiosis. Because one cell is living inside the other. Now, starting off with animals, this is easy because there are a number of animals which perform photosynthesis. The spotted salamander is the only vertebrate known to do that, as far as I know. But for example, corals. Corals are animals which perform photosynthesis. How could animals perform photosynthesis? Because they have algae living inside them. There are different kinds of algae which can do this. In this case, they're called dinoflagellates. But our coral reefs depend on algae, dinoflagellate algae, which live inside animal cells. So what are called zooxanthellae in coral allow these little coral animals to make these great reefs. And reefs are then important for marine life. And so there are many examples of this. I gave some with animals, but when it comes to, say, euglenozoans, for example, most euglenozoans have to be, either predators or parasites. So, for example, here's, this purple euglenozoan. This causes sleeping sickness. It's what's called a trypanosome. So, any second now. So that's a parasitic cell living inside, human blood. But some of its cousins are green and perform photosynthesis. How can they be green? Because there are cells living inside other cells. And so the first of these euglenozoans, and the majority of them, do not have the ability to perform photosynthesis. But euglenozoans can have cells performing photosynthesis inside. So cells inside other cells. And I can go through many examples of this. So diatoms, dinoflagellates are examples of cells which exist inside other cells. So that's possible for cells to live inside other cells in a relationship where they both benefit. And the reason that I'm mentioning that is because not only is that possible, that it occurs more recently, say with spotted salamanders or with corals or in some euglenozoans, it seems to have occurred in the very first eukaryotes. The very first eukaryotes seem to have resulted from endosymbiotic events, which gave them the mitochondria, a feature which all eukaryotes share, although we could argue about a few simple ones like Giardia. And then chloroplasts, which I'll get back to. So if we consider a eukaryotic cell, all right, in addition to the nucleus, there could be other membrane-bound structures like mitochondria and chloroplasts, which not as big as the nucleus, but then would be the second largest organelles inside the cell. How did eukaryotes get this? How did eukaryotes evolve? Well, it seems to have been an event of endosymbiosis because there are proteobacteria which are similar to mitochondria. The cyanobacteria, the blue-green algae, are similar to chloroplasts. Why do I say that? Well, because they have so many features in common, all right? So When you compare the proteobacteria and the mitochondria in living cells like ours, when you compare chloroplast to cyanobacteria, they're roughly the same size. They have similar shapes. They divide by binary fission like bacteria, not through mitosis, and the structures which allow the rest of the eukaryotic cell to divide. The chromosomes here are circular. Now, in eukaryotes, the nuclear chromosomes are rod-shaped. Circular chromosomes remind us of bacteria. There are genomic similarities. So, for example, if you speak English to different people in the world, you sometimes pick up accents, or in this part of the world, this word means this here, or this word means this here. So, in Spanish, it's the same thing. So, everyone, say in Latin America, speaks Spanish, but this word in this country has a slightly different meaning than this word. Well, the same thing here, DNA codes. Sometimes there are a few exceptions where, while everyone's speaking the same DNA language, the nuclear genome, this codon, say, means stop. But for a bacteria and for a mitochondria, this word means put this specific amino acid in place. So mitochondria speak DNA speak with a bacterial accent, not with the accent of the nuclear genome. The way that translation and the making of proteins is initiated is similar. And mitochondria and chloroplasts can have similarities with ribosome subunits, porin proteins. They can have their own plasmids. These are extra chromosomal loops of DNA found in bacteria. And then antibiotics. So for example, I can't take high doses of penicillin. I'm not allergic to penicillin, but nobody can take really high doses of penicillin. Why? Penicillin kills bacteria. And inside my cells, I have mitochondria, which look like bacteria and act like bacteria. The antibiotics which kill bacteria in high enough doses are damaging to our cells because our cells contain things which look like bacteria. Those who are allergic to penicillin, it's because their mitochondria have a variation which makes them even look more like bacteria than the typical. So not only does endosymbiosis occur from time to time, it seems to have occurred at the base of the eukaryotes. Eukaryotes have mitochondria as one of their shared features. And mitochondria seem to have arisen from bacterial cells living inside larger cells. So endosymbiosis helped give birth to the eukaryotes. As we can discuss later, the same is true of the cyanobacteria. So green algae, they're green. They perform photosynthesis and they release oxygen. Plants, which evolved from green algae, are green. They perform photosynthesis and they release oxygen as a waste. Why? Because inside green algae cells and inside plant cells are the descendants of cyanobacteria, which are green, perform photosynthesis, and produce oxygen as a waste. So the chloroplasts, which we associate with plants and green algae, are actually the descendants of blue-green algae, the cyanobacteria, which evolved these features. So for an example, our cells have mitochondria, and they can have lots of mitochondria. I mean, a big skeletal muscle cell, I mean, just got thousands and thousands of mitochondria. And mitochondria are the size of bacteria, the shape of bacteria, but they have their own chromosome. So while humans have 46 chromosomes in their nucleus, they have another one in their mitochondria. It has its own genes. Sometimes we get sick because of mutations in our mitochondrial genes, not in our nuclear genes. All right, so humans with their eukaryotic cells then have features, including endosymbionts, which go back to the origin of eukaryotes. Now, I would like to make what could be a very long discussion, very short, because these first eukaryotes then diversified, and today there are so many lineages of these protists, but I would like to just kind of bring it all together for the the history of life. So, for example, there's Giardia. which is possible intestinal parasite, one of the reasons we treat our water. This actually may be kind of one of the first branches on the eukaryotic family tree. So if we were to picture all of the eukaryotes here, Giardia seem to be an early branch, given some of the primitive features. So they have mitochondria-like structures, but they're the only ones which don't really have mitochondria. So it seems like after that first endosymbiosis occurred, Giardia are a lineage which broke off before that endosymbiont became mitochondria. Here you can see lots of little paramecia swimming around and a big amoeba which is feeding on these. So then both of these are what are protists. So they are eukaryotic cells because they're bigger than bacteria. They have the nucleus, etc. But you can see how diverse they are. So protists include amoeba, that's one lineage, and they include ciliates, that's another lineage. And when I say lineage, I don't want you to think it's just a couple that are kind of like, I don't know, like let's say like the dog genus Canis, or the dog, family Canidae. These then are entire groups which can be broken into classes and orders, etc. So there is an enormous number of, say, ciliates. So ciliates are a group of protists. So just look at some of the diversity of these ciliates. So you'll see that there are familiar ones like paramecia, but there are some which are much larger. All right, and actually are visible to the naked eye. You can see this without a microscope, and some actually resemble worms. They're not worms, they're one-celled big. And so there's all of these different kinds. There are some which I will get back to because they're helpful in the fossil record. There are dinoflagellates. I'll mention the diatoms again. There are a number of groups which seem a little more similar to fungi. Then there are the algae. Now, algae, there's certainly different kinds. So we have green algae, brown algae, red algae, golden brown algae. So there's lots of different algal groups, some of which can be quite complex. Brown algae, for example, include kelps, which can be well over 100, 150 feet long. So protists, some of them are unicellular, some of them are much larger and can vary a great deal. So point being made is from these early eukaryotes come the diverse lineages which are alive today. And the term we use for the group called protists is it's not a great biological group. It's what's called a paraphyletic group, because from these different lineages, we get animals, plants, and fungi. So once again, if we were to put the family tree of the protists, notice that how big it is. And so there's like algae is not one thing. So brown algae are a kind of algae. Diatoms are algae. Some of the euglena perform photosynthesis and are algae. But the red algae are over here, and the green algae over here related to land plants. So there's all of these different lineages. Fungi arise from some groups of protists, and animals are related to a group of protists called the choanoflagellates. And so therefore, protists are what we call paraphyletic, because while amoebas are descendants from the first eukaryotes and are still called protists, animals are descended from the early eukaryotes, but they're not included in protists. Plants evolved from the early eukaryotes, as did fungi. So protists are eukaryotes, which don't include some of the more complex descendants, such as the animals, the fungi, and the plants. And so as such, it isn't the best biological group, because notice that while the term Protists includes many of the descendants of the first eukaryotes, it doesn't include these. Now, getting back to the fossil record, some of these protists fossilized, especially those which would make hard shells around themselves. And I'll get to some of the ones which can do that in the modern world. But when you look at fossils of microscopic life, there are many forms which can help you identify fossil strata, even potentially locate oil. So they're important. There's geologists who are studying fossil rocks, looking for minerals and petroleum, et cetera, often do something known as stratigraphy or biostratigraphy, where they say, you know, this is the rock layer that we're looking for, and here are some organisms which can help us find this rock layer, find the layer where petroleum reserves, for example, tend to be deposited. So let's look for these. So what you're seeing are microscopic organisms which are extinct today, and nevertheless, they are very important very often in studying rocks, because some rocks will have one kind, some rocks will have a different kind. And we lump a lot of these together in with the term acritarchs, and study the world's ecosystems this way, because after studying them, one realizes, well, some of them were found more in cold water, some were found in more in warm water, that there were mass extinctions, and one can tell not only would like some of the animals disappear, but also then some of these, and so different types of prokaryotes, or I'm sorry, different types of these protists, these primitive eukaryotes, they lived at different times. So some of these lived in the early Neoproterozoic, some lived during the Ediacara and the Ordovician, and so you can identify time periods by which of these were alive at the time. Now, what are these? Well, because they are one-celled, and because we don't have them to truly study, we just kind of have their outlines, there is some disagreement, and they're probably a diverse group. So are they green algae? Are they ancestors of the Dinoflagellates, which are alive today? Are some of them fungi? Are some of them animal embryos? Are some of them advanced cyanobacteria? Because they can get to be pretty big and complex. Or is it a mix of those? So acritarchs, which are named and described and used for geological purposes, many of them don't necessarily have to be closely related to each other. All right, and so some could have been the embryos of animals, while others are primitive fungi, while others are green algae, etc. All right. So as the first eukaryotes arise 2 billion years ago, all right, and the acritarchs, this collection of eukaryotic cells, they start then to increase in the Mesoproterozoic eon. And then one of the ways we can, study some of the, eons of the late Proterozoic is by looking at these acritarchs and looking at their diversity. But then even once, the Paleozoic era starts, we can look at, at certain times the acritarchs increase in diversity, and then later some groups would disappear. Was that tied to geological events? Was it tied to animals which were filter feeders and preying on them? And so, they could help us understand the ecological implications of the ecological implications of the changing world, and at the same time when acritarchs are reaching their greatest diversity, or in the Ordovician period, animals are undergoing such a great diversification that it's called the Great Ordovician Biodiversification Event. So acritarchs, which were probably food for many filter feeding animals, were they then important then in the evolution of animals. So some people study these fossil protists because, once again, they are once again, they're important in food chains and the like, but some of them are especially easy to study. So think of, for example, a snail versus a slug. A snail and a slug, they're the same type of animal. They're both mollusks. They have the same body shape. But because the snail makes a hard shell around itself, it's a lot easier to study snails. You can go to the seashore and collect snail shells, but slugs harder. In the same way, look at these shells. These are calcium carbonate shells of microscopic organisms known as foraminiferans. There are beaches where someone says, oh, look at the wonderful pink sand beach here. It's not really pink sand. It's the cytoskeleton, it's the outer casings of these foraminiferans made of calcium carbonate. So some microorganisms form these shells around themselves. So foraminiferans are an example. Radiolarians are an example as well. Instead of calcium carbonate, they're using silica. So there would be an amoeba-like animal living inside, these casings. But some paleontologists now, it's easier to study these because just like shells are more likely to be preserved while the soft body of the animal is likely to be lost. The same thing is true here. You can study then the I'm sorry, I'll do the other one. You can study the casings, which are more likely to be preserved. So here, these foraminiferans and radiolarians, there would be like amoeba-like structures that could leave them in life, but we see the remains of them, they're hard shells. There are other groups. Dinoflagellates have hard casings around them. So do diatoms. Diatoms, like the radiolarians, their casing is made of silica. You can see how glassy it looks as opposed to the calcium carbonate of, say, foraminiferans and others. Now, this fact that a lot of the one-celled organisms then make these hard outer casings is then interesting because now there are paleontologists who specialize in this. So these casings are easier to find fossils of than like a soft-bodied amoeba. And so these casings include fossil organisms. And so some individuals are using these for stratigraphy, where they'll say, oh, look at these diatoms, or look at these radiolarians. One can see the difference, and one can say we find petroleum when these were laid down at this time period, but not so much when these were laid down. So many paleontologists are not digging up dinosaur bones, but instead they are looking for microscopic life. And when we consider then the diverse rocks, many rocks then are composed in part by these microscopic organisms. Some limestone, all right, is made of calcium carbonate. And so those foraminiferans and other microbes, they can contribute to to limestone. And so in the fossil record, we find limestone, and microorganisms contribute to that. A group which is still alive today, but more common in the past, known as the coccolithophores, they contributed to chalk. All right, and so I think half of the world's chalk was made during the Cretaceous period by these coccolithophores. Chalk, the word Cretaceous actually is derived from the Latin word for chalk. Chert is a mineral which, instead of being made of calcium carbonate the way that limestone is made of silica. All right, and so things like diatoms and radiolarians can contribute to silica. So these one-celled organisms, they can be very important in rocks and the study of fossils, but they also have given us many of the the different types of rocks. And so if you're finding chert in a specific area, or chalk, or limestone, then that gives you information about the microscopic life which was there. Okay. Two final points. One is, when we think of sex, I think most people would then think of animals. All right, so animals have reproductive systems with organs, they have courtship behaviors, and there is so much, sex that it's all around us. I mean, when you ask why these ducks have this, pretty coloration, it's so they know who to have sex with. When you ask why are these birds making these calls? Why do we hear, frogs making calls, or lightning bugs, lighting, and crickets and the like? It's for sex. Well, a question is, well, why is that? All right, so why is there sexual reproduction? Why is it so common? And that's relevant to this lecture because it was the eukaryotes which invented sex. Although sex is obvious that in animals, plants have sex, fungi have sex, and then these eukaryotes, protists, etc., they have sex. Bacteria don't. So eukaryotes, they invented sexual reproduction, and the sexual reproduction of plants, animals, and fungi is then derived from the sexual reproduction of the early eukaryotes. So first, I'd like to kind of explore why eukaryotes have sex, why sex would have evolved, and then just kind of wrap up this topic of early eukaryotes. Why is there sex? Well, interestingly, there's certainly advantages to not having sex. So bacteria, they're the most common living things on Earth. They don't have sex. And if you could take a bacterium that reproduces every 10 minutes, give it 24 hours. If you gave it enough food and took away its waste, after 24 hours, you would have so many bacteria that the mass of progeny would weigh as much as planet Earth. And after several days, then you would have more bacteria than there are protons in the universe. So exponential growth is something that asexual organisms are capable of. If you were to compare, say, sexually reproducing organisms and asexually reproducing organisms, the asexual ones reproduce more quickly. That's an advantage. Some eukaryotes can reproduce asexually. In fact, these euglenozoans, as far as we can tell, they never have sex. So while they're eukaryotes, not all eukaryotes have sex. Some have lost it. So euglenozoans no longer have sex. Dinoflagellates no longer have sex. They are just asexual. And that's one of the advantages, that it lets their population grow more quickly. They can be isolated and still reproduce. So let's just use animals as an example. If animals, say, find themselves on the opposite side of a mountain compared to the rest of their species, or on the other side of a big river, or if they make it out onto an island, if they're sexual, they are then celibate for life. They can't procreate anymore. Whereas if they are asexual, they can still divide. So if one of these were to end up in its own pond, it could still, reproduce and found a new population, whereas a sexual organism could not. In sexual reproduction, then your offspring might be at a disadvantage, not because you had bad genes, but because your partner did. Whereas if you're asexual, then your progeny have only your genes. So there are advantages to not reproducing sexually. There are advantages to asexual production. So for example, you can be isolated and still reproduce. Your partner wouldn't contribute bad genes, and the population can grow more quickly. But there certainly is sexual reproduction, which then almost begs the question, why? If there are advantages to asexual reproduction. Well, interestingly, in addition to there being advantages to asexual reproduction, there are disadvantages to sexual reproduction. So for example, if now there are two different genders, we might use the term mating types when we deal with other organisms. So paramecia don't really have genders, they have mating types. There may be more burden placed on one than another. So these are female snapping turtles, which have traveled far from their ponds to lay eggs, exposing them to risk, predators, etc. These female bison not only had their young develop inside their bodies, they underwent pregnancy, which was a time of decreased mobility for them, perhaps increased risk of predation. They invested a lot of energy. And then in mammals, obviously, there's even energy invested after birth with the production of milk. Now, males don't have the same nutritional investment. And so the females in sexually reproducing organisms may find themselves investing more nutrition, and that's less for them to make it through the day. Now, if two individuals are going to reproduce sexually, they have to find each other, potentially at least. And so many animals, the way that they do that is that one of the genders, typically the males, then are more noticeable. They're more brightly colored. They make a courtship song, etc. And so here you see birds making, courtship songs, but these are the males. The females are less brightly colored, typically, and they are not making these courtship songs. Now, that certainly, allows for sexual reproduction. So, at some point, you should.